All right, our next presenter is Adam Riesselman from Harvard University. His field is bioinformatics and integrative genomics. His advisor is Deborah Marks, and he did his practicum at Lawrence Berkeley National Lab in 2016. So today he'll be presenting on predicting the effect of mutations with generative models of evolutionary sequences.
Great, thank you very much. My name's Adam Riesselman, and today I'm going to be talking about generative models for genomics. First, I'd like to give a big shout out to a fellow graduate student, John Ingram, and my PI, Deborah Marks, for helping me so much on this journey. You know, this wouldn't be possible without them. First, I'd like to give a basic tutorial, kind of intro to molecular biology, just to get everybody on the same page. And that starts with DNA, the instruction for all life on this planet. We have about 3 billion base pairs of DNA in our genome, and it is encoded into 4 different bases, C, T, A, and G, as you can think of it. And the central dogma states that this DNA is translated to RNA, and this RNA is turned into protein. And these are the molecular workhorses of the cell, as I'd like to say. This is a chain of 20 possible amino acids, and depending on the biochemical composition of the amino acids and their ordering, can really change the property of this molecule. This protein, which is a chain of amino acids, folds up into a complex 3D structure, and depending on the structure, will determine its function. So this function can do pretty much anything in the cells. They're the things that digest the food and take the energy in that we eat. They take light in, and our eyes use proteins to see what's in front of us, and proteins that, you know, the light from the sun takes in for plants to turn it into energy. They're the things that do stuff in the cell. So one of the questions you might be asking, and that gets asked a lot in biology, is that if you make just one little change to this code inside of a protein, say in this case glutamic acid, which is represented by an E right here, and change it to a leucine, which is an L, how's that going to change the function? In the case of this protein, beta-lactamase, which is implicated in antibiotic resistance, how is that going to make a microbe grow in the presence or absence of some antibiotic? Now, you might ask, well, say we mutate that position to another amino acid, so this glutamic acid to an isoleucine, which is a different amino acid with somewhat similar but different biochemical properties. Is the effect of that mutation going to be the same? Is that function the same or different? And this problem pops up all the time in biology. So for in disease, if a patient walks into a clinic, they're sick, and we want to understand this disease, one might ask the question, is this mutation causing cancer? Say if we want to treat this cancer with a biologic, say an antibody or something from the immune system, we might ask, does the sequence of this antibody cause this antibody to be stable in the patient? Say we're trying to start a company or something like that, and I'd like to have some protein that digests wood better to make biofuels in some hot reactor. We could ask, will mutations in this protein that digests food make it better or worse? But this is a really hard problem. So what people do, the current state of the art, is that you start with a set of sequences, you may have heard this before, and you change them to all possible amino acids. This is just showing at one position, but you could do it across the entire sequence. And then you do some selection. This is kind of some like laboratory evolution, you could say. And that like tests some function. So for the example of beta-lactamase, it'd be asking like how well does this thing resist an antibiotic? And then you count the number of instances before and after selection. So what you see here is that these were all pretty homogeneous before selection, then afterwards the H's and the Y's were preferred. So we'd say these mutations are tolerable, they're functional. Everything else probably caused that organism to die. It wasn't fit, so it is a deleterious mutation. So you just compare the ratio of sequences before and after an experiment. So you can do that for all possible positions in a given sequence. So for example, in this protein, it's a PDZ domain, which it's a protein that binds other proteins. You can mutate all the positions to every possible amino acid. And if in this figure, if it is white or pink, the mutation is fine. It doesn't really change the function. But if it's blue, it essentially breaks the protein, and it doesn't work anymore. So understanding the effects of mutations is really, really important for biology, but it's really, really hard. So for like any given protein, if you want to mutate 1 or 2 or 10 or 20 possible positions, you'll have to do maybe 100, tens of thousands, hundreds of thousands, even millions of experiments just to probe at the function of one protein. And this, as you can imagine, is pretty slow. Doing this experimentation can take months to years. And it also is pretty expensive. You have to order the reagents, you have to do the experiment, you have to transform it, and this can take a lot of time and a lot of money. So there just has to be a better way. And since we're at the CSGF, we know, of course, it's with computers. So I'd like to take a step back and kind of think of another way that we could do this. So this is like a generalized approach. So this is like protein X. This could be your favorite protein. Humans have about like 26,000 of them, and there's lots in all these other different organisms. But there are lots of other examples of a given protein out there. So if I've asked you to sculpt, a very specific sculpture, what you'd probably do is you'd go out and ask for all possible examples of that similar sculpture. So we can do the same thing. Say we're interested in some protein found in humans that is like central to the metabolism. We pull that out, but then we also know that monkeys have the same protein, just slightly different, but it does the same function. Oh, and also mice have it too, maybe a jellyfish even, or like a plant that all have these same sequences. So these are all functional, homologous examples of a given protein. Homologous meaning that they share the same structure and function. So there are lots of public sequence databases out there that have a lot of genome data sets. So like a big shout out to JGI, which is sequencing oodles and oodles of organisms. These sequences are deposited in public databanks, and we use them, and it's great. So the way I like to think about it is that natural evolution is an experiment in itself. If I see a mutant in one of these databases in a sequence, I would say that if it's present in the database, it's probably a tolerated mutation. So you can imagine that all organisms are mutating, they change, and then if the mutation is good, it will have children or little, small microbes, and it'll propagate and be recorded in the sequence literature, or like kind of sequence databases. But if it mutates and that mutation is deleterious, that organism dies out. And so it's depleted. We never see it before. So we just need a good way of getting this data and summarizing it down. So what we can do is we can find all possible examples of a given sequence, and then we can organize it into what I'd call a multiple sequence alignment. So what you do is that each of these lines is a different example of a sequence, and each row is a position with a shared ancestry. So this is a way of just kind of vectorizing your data. And so each of these positions would kind of have the same structure and function in like a crystal structure. It's just one point. So we can get lots and lots of this data. So what I'm showing right here is this kind of in color form. Imagine each one of these pixels is a different amino acid. A row is an organism, and a column is one of those shared positions of ancestry that I talked about before. And we can get, for like a really basic enzyme, we can get lots and lots and lots of examples of this. So I'm going to propose like a general way of kind of like a cookbook for making these kind of probabilistic generative models. So we take this multiple sequence alignment, a bunch of examples of the protein that we're curious about, and we're going to fit a generative model of these sequences. So what we're trying to get at is that this generative model will capture functional constraints of that proteins. We can kind of boil down what makes that sequence a sequence, and if we have parameterized our model correctly, we will capture functional constraints. So this is kind of like some unsupervised machine learning you can think of your like a set of related sequences, for example. So this is the general formula that you can take. Step 1, you're interested in any protein. Go out, find examples of it. Step 2, compute the log ratio for each mutant. So if I fit a model to evolution, then now I'm going to the lab. What I'm going to ask is, does this mutant in the lab look like something that I've seen in nature before? If I've seen it before and like the log ratio is relatively small, it's probably like the mutant is tolerated. But if I've never seen that mutant before as parameterized by this generative model, i.e. this log ratio is large, it's a deleterious mutation, and it can be kind of classified as such. And so why would we use this? It uses public data. It's effectively free. It's fast. It works pretty much on almost any protein, and it's pretty accurate, as we'll see. So this is a cookbook for lots of ways that you can do it. The first way would be just like a site-independent model. So you look at any given site, you kind of average how frequent a given amino acid is. And so like a probability of a given sequence is just multiplying some site-independent factors. And so if you just like look down this column, an R to a K, so an arginine to a lysine is neutral, because we've seen it before. But an arginine to an isoleucine, we've never seen it before in that column. It's probably not very good. Something that took that my PI got famous for and is used quite a bit in the field is a pairwise undirected model, which I'm sure lots of you folks have already talked about and are pretty familiar with. And it's a Markov random field, or an Ising model, or a Potts model, or otherwise multinomial logistic regression. And then you're setting site and pairwise terms for any given sequence in that multiple sequence alignment. And so what that looks like is that parameterized in here are the preferences for the sites and pairs. So if we have a site and pairwise model, why should we stop there? So why can't we go higher order? And we know that this is really important in evolution. This is called epistasis in the field, and there's a lot of interdependencies between sets of positions. So we're able to use kind of a variational autoencoder or a neural network powered latent variable model. You have a latent variable, it goes through a nonlinear mapping, and it generates a distribution around a sequence. So this is pretty intractable because you have to account for all possible latent variables represented as Z for each sequence. But some tricks that were developed pretty recently with the variational autoencoder are able to to solve this problem. So you have an approximate posterior, which is also powered by a neural network. So given a sequence, generates the latent variable. That latent variable is then transformed back into a distribution around the sequence. The objective function, the P of x, we set a lower bound on it with the elbow, the evidence of the lower bound of the marginal likelihood of the data. It's two terms, the reconstruction cost, I like to break down, and the information costs of the latent variables. So here we're watching it train. So for a given multiple sequence alignment, each sequence has an individual latent variable that it's associated with. And so this is of a whole multiple sequence alignment. And as what you see is that it can find some structure of relatedness in the data as it's solving, which is pretty cool to see that it's finding some order in the data, which we know is there. We found that our model actually overfit. So we made this the generative model, what could be called like a Bayesian neural network. So we have so the final objective function that we solved, we have the reconstruction cost around the sequence. We have the information cost of the latent variables, as well as the information costs of the parameters of the model. So that's the cookbook. Infer a generative model of a sequence family, and approximate the log ratio with the elbow. So what we did is we went out and from the literature got 30 saturation mutagenesis experiments. So we got ground truth of what the effects of mutations are in the lab or in the real world. And then we computed alignments and calculated a probability model of those sequences. And then we compared those with the rank correlation coefficient. And here is like the Spearman rank correlation coefficient. And we can see in a large number of proteins, from antibiotic resistant proteins to proteins that are implicated in cancer, as well as pretty much all different organisms, we're pretty good at predicting the effects of mutations. And we're more predictive than both the site-independent and pairwise model. And this is, again, showing the same kind of data. Above the line is good, and our model is better. So can we interpret what this model is doing? So what we did is we built in priors that kind of take into our knowledge of biology. So this is the decoding process. We have a latent variable that's mapped through some nonlinear mapping with weights, creates a hidden vector, and then it generates a distribution around a given site. So we parameterize this, this W, or this line right here, via this. It has two parts, which I'll go through real quickly. There's a dictionary that's shared across all positions. So we know that amino acid composition is shared across the entire sequence. So we should share that information for the whole protein instead of just restricting it to a given site. And finally, we also have a scale parameter across a given activation. And what this does is it is like a sparse gating mechanism that allows sparse interaction patterns for a given activation. So what we can do is, if we first look at this dictionary parameter, we take the correlation coefficient of like the dictionary parameter across all of our different models. It pulls out biochemistry. We're learning like evolution. We're learning from evolutionary data. And without us giving anything, it's able to pull out evolution, so in amino acid preferences. Also, we have the sparsity parameter. We can map that back onto the crystal structure. And we find that it's co-localized in 3D on the crystal structure. We didn't have to put any information of the crystal structure in there. It finds it itself in the data. And that was really exciting. So just to recap, predicting the effects of mutations is really important for a lot of places in biology. Generative models are really good for kind of approximating what the effects of mutations will be in the lab. And latent variable models predict better than this, or are better than the state of the art for predicting the effects of mutations. So I'd like to give a big shout out to the DOE CSGF. It's really impacted my science and has made me so much better scientist. And I'd like to thank all you guys for that, as well as Harvard, Debbie and John, Chris Sander, and everybody else in the Marks Lab. Thanks, and I'll take any questions.